of pseudonychia and domoic acid toxicity on razor clams on the Western Coast. I'm doing this project with Dr. Vera Trainer, who is the director of the Olympic Region Harmful Algal Bloom Partnership, and Dr. Chris Free, who is a professor at the University of California, Santa Barbara. So first I want to talk about toxic harmful algal blooms. They are the result of rapid and excess growth of biotoxin-producing algae, and this can accumulate in seafood species, especially filter feeders. These events have increased in both frequency and intensity for the past few decades, with previous literature pointing to causes being rising ocean temperatures, water column stratification, eutrophication, ocean acidification, just kind of, I guess, everything going on. And during these toxic harmful algal bloom events, commercial, recreational, and subsistence fisheries are interrupted. And the species we'll be focusing on today is pseudonychia. It produces domoic acid that can accumulate in filter feeding shellfish during blooms, or some of you might hear them as red tides instead of harmful algal blooms. Consumption of contaminated shellfish causes domoic acid or amnesic shellfish poisoning, which includes symptoms of vomiting, diarrhea, nausea, and amnesia, so a little short-term memory loss. So it starts with pseudonychia, and they will produce domoic acid. Again, it gets into the shellfish, and then through consumption, we can get these symptoms. Rarely death, luckily. And the shellfish I'll be focusing on is razor clams. They're one of the largest clamming fisheries in Washington. They're valued at around $40 million annually, including for like tourists and recreational passes and such. And it's a focus today because they accumulate domoic acid particularly fast, and they can keep it in their tissues longer than some other shellfish. Across all bivalves, there's a limit of 20 parts per million of domoic acid. And again, during these bloom events, we have tribal, recreational, and state fishery harvests paused until it's below the limit. Because of the increased frequency and intensity of harmful algal blooms, the Olympic Region Harmful Algal Bloom Partnership was established in 1999. It's a partnership between the Washington Department of Health, commercial, tribal, and recreational fisheries along the Washington coast. And to provide early warning for these toxic bloom events, they use pseudonychia and particulate domoic acid as early warning. And through visual analysis, they established thresholds of small pseudonychia being about 30,000 cells per liter, and large pseudonychia being about 1 million cells per liter in 2005 to indicate early warning that there should be a toxic event soon. And they also established a limit of 200 nanograms per liter of particulate domoic acid, which you can test for in the seawater. Oh wait, that's my answer right there. So how this is kind of different from before. Before you would, it was expensive and time-consuming last-minute closures that result of you were digging for clams, you send us off to the Washington Department of Health, and they test the tissues for domoic acid toxicity. But now we can collect plankton samples and look for toxic cells under the microscope and get a general count of how much is in the seawater. And if it reaches above this threshold, then we will start using toxin tests in the seawater to see if we reach the threshold of 200 nanograms per liter. And then if both those are up, then you do more testing of clams at the health agency. And monitoring toxic pseudonychia is not straightforward. So I talked about small and large morphotypes. This is because there are different species of pseudonychia, as it's a genus, and not all of these species are toxic. So the ones highlighted right here, these are toxic, and you can see there's different, fairly different morphological traits, which is why we separate them into small and large, or they did in 2005. And I will continue to use that. So my questions were, do the current particulate domoic acid threshold of 200 nanograms per liter provide adequate warning for these toxic blooms? And same for the current thresholds of the small and large morphotype. And I hypothesized for both of these that there would likely need to be a lower threshold for both due to changing ocean conditions. So here's our first site map. This is the Washington coast. We have these different management zones that were created by Free et al. In a 2023 paper that just allows for easier management and closing of various locations and testing. So I wanted to have that represented here as well. And the site, the project site focus will be on Long Beach over here, just because not enough time to look at all of these. And then this is the same sites for razor clams, and there's multiple sites for razor clams at Long Beach and Douglas Tropic Shore tested, but I did a daily aggregation average of these for our viewing pleasure. So first, here's an overall of razor clam toxicity in the past about 25 years on the Washington coast. Any sort of dot you see, that means it's over the limit of 20 ppm, and the size represents larger toxicity, and then the color, again, percent over the threshold. Here are the sites we'll be focusing on right here. You can see there's a large bloom event about 2005, 2015, around 2017, 2018, and another around, I think it's 2022 or 23. And so we have a lot of information on these graphs we'll be looking at next. So I'll take my time and kind of walk you through them. This first graph over here is Pseudonychia abundance. The dark red is representing the large morphotype, and the light red is representing the small morphotype. And then you also see these dashed lines down here. Those are the threshold values for the respective morphotype. In the middle, we have particulate domoic acid. It is in log scale, and the dashed line again represents the threshold. And on our graph on the right, we have domoic acid contamination in razor clams. Again, the dashed line representing the Washington Department of Health threshold. And we can see in these years, first the threshold was reached for Pseudonychia large at around April, May. And we also see during that same time increase in particular domoic acid contamination. And again, at the same time, we see higher domoic acid contamination in razor clams. So it didn't really provide us much early warning, but it was accurate in that it did catch the toxicity in razor clams. But also later, again, you see larger spike right here and a spike in PDA, both over the threshold, but we don't see domoic acid contamination in razor clams increasing. It's because we talked about earlier, this is a mixed assemblage of species, so they're not always toxic, and we can't necessarily tell, which is why we also include the particular domoic acid. But again, sometimes it doesn't work out necessarily. Here's another example, looking at 2019, and this shows, again, false positives. We see that the thresholds for both the small and large morphotypes went over the threshold a few times throughout the year, but we did not see particular domoic acid reach above the threshold. And during the same time, the domoic acid contamination in razor clams also didn't go over the threshold. And these false positives are much preferred over the Washington Department of Health versus being unprepared. In 2020, this is a good example of the early system working. So in May, June, and August, we did see an increase in Pseudonychia, but it did not result, and then this resulted in us starting to test for particular domoic acid. Sometimes they'll wait to test just if they see larger cell values. Other times they'll just do it weekly for cost effectiveness. And this provided adequate early warning for a toxic bloom event that was going to happen around here. It's October, November. So you can see it went over and it starts to increase, and then it goes over again around that same time, and where all the thresholds are surpassed. In 2022, we see, again, this is during the growing season where you'll often see June and July, August, they'll have the highest cell abundances. That we see the Pseudonychia, again, reach over the threshold, mostly for the large morphotypes, some of the small morphotypes as well, and again with a particular domoic acid, but this is only increasing domoic acid contamination, and it hasn't yet reached the threshold for you can't consume them. So this provided a few months early warning, which is really good. Yeah, there you go. In 2023, we see something different. Especially the large morphotype is well above the threshold. It's all the way down here. But we do see particular domoic acid, again, also go well above the threshold. I'm going to remind you, this is log scale, so about 1,000, and this dashed line represents 200. But we do not see high contamination of domoic acid in razor clams. So, and something else I want to point out is that these razor clams can retain domoic acid for up to a year, so they can kind of retain domoic acid from previous events. Another bloom can spawn, and then that will can rapidly increase domoic acid content. And so kind of revisiting my questions, do we need different thresholds for both pseudonychia, small and large morphotypes, and particular domoic acid concentration? I think the current thresholds still fare well, though you can see false positives do still occur. And I think just getting more years of pseudonychia abundance for the small and large morphotype and the particular domoic acid concentration could be used to help better investigate this, along with looking more of the Washington coast and not restricting to Long Beach. And then some other future directions for this work is using pseudonychia abundance, particular domoic acid concentration, and razor clam toxicity to provide early warning for Dungeness crab. I did do a bit of this for this project, but I haven't gotten enough time to finish that part. So preliminary results do show that these three parameters can provide early warning for Dungeness crab because they are offshore and they do come onshore and they can feed on to razor clams and other contaminated bivalves. And I would like to thank my mentors, Dr. Vera Trainer and Chris Freed, for helping me, especially Chris with the R programming, and Dr. Vera Trainer for just helping me interpret these graphs. Also the School of Aquatic Fisheries Sciences and the Leo Maddox Foundation from the School of Oceanography for supporting this research, along with the many tribal, commercial, and recreational partners that ORF has, and the multi-stressor project partners, which is part of a larger project for looking at the multiple stressors on West Coast fisheries. Some sources. Any questions? Yeah.
Is anybody looking at the possibility of eDNA, environmental DNA, to identify which species are actually being found? Because if some are toxic and some are not toxic, it doesn't really matter how many there are.
Yeah. Yeah, exactly. That's why they use the particular domoic acid, but there definitely, I think I've read some studies where they are trying to use eDNA. There's also this newer instrument that I'll be working on in my PhD program that takes pictures of the plankton and can identify the species through machine learning. So I think that's another way to go. Yeah.
Are clams infected with domoic acid only dangerous to eat if you eat them raw, or can you cook them?
You cannot cook. The domoic acid is a very stable compound, so you cannot cook it out, unfortunately. So these just have to be tossed. Yes?
Okay, one of your early slides, how was the domoic acid analyzed? And you showed something that looked like an antibody test with
Yeah.
sort of what they were using for COVID, for example. Is that used on water, or is that used on?
Yeah, so this just generally gives
the test for the toxin there.
Yeah, so this is just like kind of like a quick test just to see how if the water is like fairly toxic, but they do take it back to the lab, and I forget exactly how they analyze it, but they do bring in like a liter of seawater and do it that way. But this is just kind of like a quick test that you can do like at the beach in the water.
So where are they taking the water samples? 'Cause, I mean, the razor clams are right on the beach. And
Yeah.
I'm just wondering if some of the disconnect between toxicity in the razor clams and the presence of the other berries because of whether they're actually, whether the pseudonychia is actually on shore or not, or if it's just offshore.
Yeah, they do onshore testing. So I got to go visit earlier this year. We just kind of walked onto the beach, and grabbed a bucket of water, and that's one way. And that's how they test for the pseudonychia abundance and particular domoic acid. And they do have less sites for that than they do for like razor clam testing. As we can see, like in the same area, there's just like one for that, but versus razor clams are tested throughout Long Beach. But both tests are done onshore. Or not onshore, but taken onshore.
Okay.
Any more questions? Yes.
How much of like the time series of the domoic acid covered like aligns with the fishery season?
Wait, sorry, what do you mean by that?
Like, do we see the spikes during the crop season?
Oh, yeah. Sometimes you do and sometimes you don't. Like over here, the season's more, I think it's in spring, summer, but So like you do see it over here, and it will close, but also during like lower seasons, like over here, and I don't believe there's much collection during this time, but it was high. So it kind of just vary throughout the year. It can be hard to pin down. It can happen multiple times a year as well. So.
Thank you.
Thank you.